Okay, thanks, Steve. So I'm going to apologize for two things in advance, one of which I already apologize for, which is that I have to leave tonight because I'm starting a new job on Wednesday, and I moved my lab on Thursday of last week, and everything is like chaos, so I have to get back to it. And the second is that you're going to have to listen to me talk for the next 3 hours, because this is the only way the schedule would work, so that I could say everything I wanted to before I left. So the sample network part that Steve mentioned is going to be the second of my 3 talks. I thought I would start in the first one with a sort of catch-all category about designing a gene co-expression study, and just try and sort of encapsulate some of the things that I tell people, when trying to advise them on designing their own experiment to examine gene co-expression relationships in their favorite biological system. I think that some of the points and themes will be applicable to other kinds of data, beyond just gene expression data, or at least I hope they will. But the reason that I'm interested in gene expression primarily is because, actually there's several reasons. The obvious one is that biologically, gene expression is necessary, although not sufficient, for most biological functions, right? And also, more practically speaking, the technologies that exist for measuring gene expression, namely microarrays and now RNA-seq, are pretty mature, right? What that means is that they're very precise, they give very precise data, it's multidimensional, and it's relatively inexpensive, so you can generate large numbers of samples, which is of course what we want, and there's a huge repository of information that's already available online for free, right? And in fact, during my PhD at UCLA, that's how I made my living scientifically, was reanalyzing other people's data. And I would argue that there is no other ohm that we can measure that contains as much functional information about a biological system as its transcriptome, right? It's the final common output of all the epigenetic stuff, all of the genetic stuff, and the starting point for everything that follows. And yet, historically, the vast majority of studies that have analyzed gene expression data have looked for differentially expressed genes, right? This is a heat map just showing what would be considered to be an excellent result from a differential expression study, where you have rows corresponding to genes and columns corresponding to samples, red high expression, green low expression, and you can see there's a large group of genes whose expression perfectly delineates these two groups of samples. But then, you're left with this problem, which is how do you prioritize the genes in this list for further study? You feel kind of arbitrary. So this is kind of an imperfect analogy that I came up with as I was, I'm always trying to think of analogies to explain this stuff. But, you imagine that you're thinking about 2 songs and you want to ask, what do these songs have in common? What's different between them? And so from a differential expression perspective, it's like asking whether one song has more Ds or F sharps than another, right? Which is not really very meaningful. Whereas in co-expression, we're really looking for the melodies, right? We're looking for the patterns. So that's one way to think of it. And so as Steve was saying, it doesn't make sense to compare songs one note at a time. It doesn't make sense to compare biological systems one gene at a time, right? Genes don't operate in isolation, so why would we treat them that way? So what this implies and sort of requires is that co-expression relationships exist, are reproducible, and biologically meaningful. And you can show that in many, many different ways. This is just one way, where on the left I'm showing basically density plots for Pearson correlation coefficients from 3 data sets. 2 are real, 1 is simulated, right? The real ones are from human brain regions. The ACC is the anterior cingulate cortex. The DLPFC is the dorsolateral prefrontal cortex. And the random is basically just normally distributed random values for exactly the same number of genes over exactly the same number of samples. And do we have a pointer or no? No pointer? Oh yeah, here we go. So what you can see is that there's an excess of moderate and strong positive and negative correlations in the real data versus the simulated data, right? And that excess is basically telling you that there are groups of genes that have correlations with each other that are higher than you would expect by chance. On the right, what I've done is I said, okay, if I just calculate out of all of the genes that I'm looking at, how many of them have at least one significance correlation with another gene, right? Where significance is defined based upon a Bonferroni corrected p-value, so a fairly conservative definition. You can see that it's about 20% in each of the real data sets and 0 in the random, right? So there's structure there that structure is effectively invisible if you're only looking at it in terms of differential expression analysis. And this is another example, that kind of brings that home, I think. And these are real data where we're looking at the distributions of gene expression levels for 2 genes in 2 different biological groups of samples, a control group and an experimental group. And you can see that neither of these genes is differentially expressed, and therefore, when analyzed through the lens of differential expression, they're completely uninteresting, right? But if you look at them in terms of their co-expression relationships, you can see that in the control group, these 2 genes are very highly correlated, whereas in the experimental group, they're not correlated at all, right? Biologically, that seems interesting to me, right? And that's the kind of stuff that you can get at systematically by looking at the data in terms of co-expression analysis, but not in terms of differential expression analysis. So, I think Steve has already touched upon some of these points, but the obvious application for co-expression analysis is just identifying the major patterns in a given data set, which a priori you would like to assume are biologically driven, but aren't necessarily so. And that's something that we all need to be very careful about, because these methods are really very sensitive to technical sources of variation as well. Dimensionality reduction is a key selling point. You go from 20,000 genes to maybe 20 eigengenes, right? That's a major gain. And it's a relatively unbiased approach. So I'll show you an example of what an unbiased co-expression analysis can tell you. This is from a collaboration with the Ophir Klein's lab at UCSF. His lab studies different biological systems, one of which is shown here. It's the mouse incisor, right? So the mouse incisor, in his view, is an excellent model for adult stem cells, because the mouse incisor continues to regenerate throughout life, unlike our incisors, or most of our incisors probably. But you can see that this is the region right here where the stem cells are thought to reside. And speaking with Ophir, he said, Mike, what should I do? This is a really poorly characterized system. And I said, well, if it were me, I would just want to get a handle on what the major sources, the major biological sources of variation are if you were to generate a gene expression data set from this system. And so we, basically, with the help of Kirsten Seidel, who's Ophir's postdoc, she dissected 94 mouse incisors, this area corresponding to the red box, extracted total RNA from every sample, and we ran Illumina microarrays, and then performed a co-expression analysis. And we did, in fact, find very interesting modules corresponding to major cell types in this region of the mouse incisor, and also related to the stem cells that inhabit the niche. But then we saw this, right? And you can see here I'm showing you a heat map, right, where each row is a probe on the microarray. Each column is one of the 94 samples. So you can see this is the module eigengene, or first principal component of the module. And then these are the top 15 genes on the microarray ranked by KME, right? So ranked by their correlation to the module eigengene. And what do you see about this module? Well, you can see that it spikes in just a few different samples, right? And furthermore, when you look at the identities of these genes, you see things like troponin, tropomyosin, myoglobin. These are canonical muscle genes, right? So what that means is this module is effectively a readout of contamination, right? So this muscle exists, just to go back a slide, up kind of here, right? And Kirsten, who's very careful, despite her best efforts, inadvertently captured some of this muscle in a handful of her samples, right? Now, to me, this is still biologically very interesting, right? Because, back to this notion of cellular heterogeneity, the prediction is that these genes are going to be expressed very specifically in the skeletal muscle cells that are driving this signature. So if you're interested in that kind of biology, this is a gold mine, right? The Klein lab is not interested in this kind of biology, so it's probably not even going to make it into the paper, right? But it's the kind of thing that you can learn from this analysis that you would never learn from a differential expression analysis. Okay. Oops. Okay, so another application I think that's important is characterizing gene functions, right? And this principle of guilt by association, which I think most of us are familiar with. Here's an example of a co-expression module. This is from human brain. These are samples from the nucleus accumbens, which is part of the basal ganglia. It's involved in addiction and reward pathways. And you can see amongst the top 15 genes ranked by their correlation to the module ligand gene, there's mostly things like RPL, RPS. Those are ribosomal genes, right? And yet this number 1 gene is glioma tumor suppressor candidate region gene 2, and there's very little known about this gene, right? And then, the question you can ask is, what are the odds that this gene has nothing to do with protein synthesis, and more specifically with ribosomal function, right? So I think that's a great selling point. Disentangling cell type specific transcriptional signatures in heterogeneous tissue samples. This is probably the area that I spend most of my career thinking about, and it all started here. Steve has already alluded to this, and this notion kind of dawned on me, because when I started analyzing gene expression data from human brain samples, the conventional wisdom was that, if you've got a hunk of cortex and you extract RNA from it, this tissue is a mix of all these different cell types. You're never going to know which transcript came from which cell. It was just going to get lost, right? And that's true, right, if you're only looking at the data in terms of differential expression. But when you think about it, if you've got 50 samples, pick your favorite cell type in the brain, let's just call it an oligodendrocyte, and just pretend that you magically knew the proportion of oligodendrocytes in every sample, and you could plot that, right? And that's what it looked like. And then you ask yourself, what would the expression pattern of a perfect marker of oligodendrocytes look like, right? So by perfect marker, you mean maximal sensitivity and maximal specificity of expression, right? So expressed constitutively in oligodendrocytes, not in any other cell types. And you would expect it to track the proportion of oligodendrocytes almost perfectly, right? A marker, with very good fidelity would show a similar pattern. A marker with good fidelity would show, maybe a little wobble, but still a pretty good pattern. And so intuitively, this made a lot of sense, right? And so when I was in Dan's lab in my PhD, I started to work with Steve and Dan to reanalyze previously published array data from human brain to test this idea. And I'll talk about that a bit more later. So biomarker identification, I think, is another obvious application. Steve has mentioned this concept of KME, which, I agree is, I think, very powerful. Here's a co-expression network produced by WGCNA. We're looking at that turquoise module there. Here is the module ligand gene, right? And here are the top 10 genes ranked by their correlation to the module ligand gene. So, as a neuroscientist, I can look at some of these genes and I can see things like CNP, NKX2.2, myelin-associated glycoprotein, and I know just on the basis of that this is an oligodendrocyte signature, right? But going back to this thought experiment, right? You can imagine if you had an infinite number of samples of tissue, right? The optimal biomarker would be the gene whose expression level correlates most strongly with the abundance of that cell type across an infinite number of samples, right? We don't have an infinite number of samples, but at this point, we've got dozens and dozens of independent data sets, right? So we can find the signature over and over and over again, and we can ask which gene consistently shows the highest KME for that module. And that gives us, in my worldview, the optimal biomarker of that cell type, right, in principle. Okay, so, some people, when I talk about this stuff, they say, what about genes that don't vary, like housekeeping genes, what do you do with co-expression analysis and that? And, I guess my answer is that there's always variation, even in the housekeeping genes. I think that's kind of an antiquated concept. This is another example. This is some of the Allen Institute data. These are samples from a region of the brain called the claustrum. And, you can look right there, and that's beta-actin, right, one of the canonical housekeeping genes. And you can see that, like all the others in this module, its expression is, approximately twofold higher among these samples than these samples. Why? Who knows? It could be differences in cellular density, differences in expression in different cell types that are represented in different proportions across these samples. In some ways it doesn't really matter. I think it just it's important to sort of let the data tell its story and not walk into it with an assumption that housekeeping genes have to be low variance or no variance. Okay. So what is co-expression analysis not good for? So I think this probably applies to more things than co-expression analysis, but it's not good at squeezing blood from a stone or trying to rescue poorly designed differential expression studies with a small number of samples, right? So here is one such example where This is like, like 10 samples. The colors correspond to different biological subgroups, each of which, no more than 3 samples and often less than 3 samples per group. And these are sorted cells with very low input amounts of RNA. And, you can find patterns, but, like you can see here, this is really spiking in one of these 2 samples that are from supposedly the same subgroup. And just given the small number of samples, I wouldn't hang your hat on this kind of analysis or this kind of result, right? Because co-expression analysis is going to find patterns of variation, whether they're real or not. If they exist, it'll find it. So here is an example of a co-expression network where we're clustering the eigengenes, right? So I know you can't read these, but these are colors. And, it's a dendrogram that shows it's clustering based upon the correlations amongst the module eigengenes. And below that are the module eigengenes themselves, right, arranged in vertical columns. So, if you look, for example, at this one here, this is what that module looks like, right? It's kind of similar to the one Steve showed in his talk this morning, and that it consists of genes that spike in exactly one sample, right? Now, do you believe that is real? No. So I think, that's why, part of the reason why I spent so much time trying to think about pre-processing, early on. I should say that many of my concerns have been mitigated as the data sets have gotten larger and larger and the technologies have improved and such. But I still think it's important, to not just, hit go on, the module detection algorithm and take at face value everything you get out, So, when you're thinking about designing a gene co-expression study, this may sound trivial, but I think you really have to think carefully about the most important biological question you're trying to answer. And that can be very diffuse, like the example I showed earlier with the mouse incisor, where you just want to characterize the major patterns of variation in the biological system, or it can be more narrow. When someone might say, well, I have my favorite genes and my favorite biological system, and I want to know what their neighbors are, right? I want to find a module that these genes are involved in. But in either case, the first thing you should do is go to the repositories, right? So there's a huge amount of gene expression data that's publicly available right now, will cost you nothing, So I always start there even now. Geo is over 1 million samples now. Array Express is also huge. That's the European counterpart to Geo. The RNA-seq databases are growing fast. You can search through these and rank data sets by the number of samples they have, by the platform, by the biological question. And I think that it is definitely the place to start. But if you can't find what you're looking for, then think about what the major biological sources of variation will be with respect to your sampling strategy. So, if you're analyzing data from different species, that's going to be a major factor. If you have brain samples from different individuals, right, then you've got all of the individual covariates like age, gender, cause of death, ethnicity, et cetera. If you're focused on samples from one individual, you can control for many of those things, right? And now, if you're talking about a heterogeneous tissue like brain, then differences in the cellular proportion among samples are going to be a major driver of variation. Things that we often don't think about, like circadian factors, right? There have been some studies that have shown that a large fraction of the transcriptome actually varies in a circadian fashion. Most of us aren't really controlling for that in our experiments, right? And then increasingly, well, I didn't put it up here, but at the level of single cells, right? What are going to be the major sources of variation when you're talking about homogeneous populations of single cells? I think that's, kind of TBD. So another point that I like to make is that we often think about co-expression being synonymous with co-regulation, and I think mentally many biologists, including myself, when we look at a co-expression relationship, we imagine those genes being co-expressed within the same cell, right, which would be synonymous with co-regulation, or at least potentially. But you look at an analysis like this, where you've got a data set consisting of samples from different human tissues, right? And this is the topological overlap matrix, and you can see beautiful modules up at the top. And when you look at what these modules correspond to, They largely correspond to different tissues. So this is, skeletal muscle with the spike in expression, heart, testes, liver, fetal liver, bone marrow, spleen and bone marrow, white matter from the brain, gray matter from the brain. So the fact that these genes are correlated across these samples, to me, says, it doesn't necessarily mean there can't be any co-regulation taking place, right? But like if I look at the genes in this neural gray module, for instance, I'll see many things that I know are expressed in different cell types, right? The reason they're part of the same module is because they're going up in brain relative to other non-neural tissues, right? Okay. Yes.
Quick question. It might be trivial, but how do you identify a particular module is from a particular cell or a given tissue?
Oh, in that case? Yeah, so this one is quite easy because, like, for example, this red stripe going down, that corresponds to skeletal muscle. There was only one skeletal muscle sample in this. It's an atlas, basically, of gene expression from different tissues. So in this case, it's trivial. Yeah. Oops. Okay. So, if you can't find a data set in Geo or Array Express that you think meets your needs and you are going to generate yourself, I think it's very important to consider the technical sources of variation that might interfere with your biology. One of them can be this contamination, which I showed you earlier. That's a sort of a biological artifact. So in brain, for instance, we often get more or less white matter in a given sample. White matter is mostly oligodendrocytes, so that can affect, the reading of the oligodendrocyte module. But that's a hard thing to control for, whereas batch effects are something that you can control for, right? And, the first 10 years of microarray studies, people didn't really know a lot about batch effects, right? There are probably a lot of published papers in which the principal findings are actually batch effects. So that's something to be cognizant of, particularly in the realm of RNA-seq and now single-cell analysis, where I think that the technical sources of variation are still incompletely understood, right? But batch effects can arise from things like having, if you're generating data as part of a consortium and you've got colleagues at one university generating data and you're generating the data, you can often see a batch effect associated with that. That could reflect, the hands of different experimenters, different reagents, different runs on the same machine or different machines. Those can all introduce batch effects. I mentioned, with new techniques, you really want to be very careful of this to the extent you can. Try and imagine what in the experimental workflow might produce a batch effect, and then balance your samples, right? And so what I mean by that is, this is kind of a trivial example, but here, you have two cohorts of samples that you want to compare, right? And there's two different experimental run dates or hide dates. And in this case, they're balanced, right? So you have both cohorts represented on both dates, whereas in this case, they're perfectly confounded, right? So I can't tell you how many published studies I've analyzed where the batch effect has been perfectly confounded with the main biological effect under investigation, not described by the authors in the study, but evidence in, for example, the header information of the Affymetric cell files where there's a timestamp with a hybridization date, right? So it's just something to be careful of. This is what I mean by that. So if this is kind of dated, right? This shows you how I got started in terms of looking at microarrays. Affymetrics is an older microarray platform, and the raw data come in the form of cell files, as some of you may know. And you can actually see right there that timestamp that tells you the exact date on which the sample was hybridized, and you can use that to reconstruct the batch information. Okay. So strong batch effects in the data often show up as the first principal component of the overall data set. And this is actually an example of the output from the sample network function that I created, which I'll discuss in the next talk. But the important point is that this is a data set consisting of serial sections through one region of one human brain called the anterior cingulate cortex. And the colors of the samples, which have been clustered by their correlation effectively over all probes on the microarray, the colors correspond to distinct Illumina microarrays, right? So each of these Illumina arrays can run 12 different samples, right? And when you look down here, right, this is a ANOVA looking at the relationship between each of these sample covariates in PC1. This one, array ID, is hugely associated with the first principal component, right? So that's a batch effect associated with the array ID, right? And that's the kind of thing that we want to get rid of. And I'll show you how to get rid of that in the next part of the talk. So, I think probably the most common question that I've been asked is how many samples do I need for a co-expression analysis? And, I never have, there's no right answer, right? You want more. The more you have, the better. But it's, obviously, and I think intuitively, the fewer samples you have, the more likely you are to observe false or spurious correlations by chance. So this just illustrates that. These are, again, just normally distributed random values. So it's a series of simulated data sets corresponding to 10, 25, 50, or 100 samples. And as you can see, when you have only 10 samples, the odds of seeing a moderate negative or positive correlation is significantly higher than if you have 100 samples, right? So as a rule of thumb, I usually tell people that it's inadvisable to pursue co-expression with fewer than 20 samples. That's just the rule of thumb. You can do power calculations, and that can help, right? And you often have to do that for grant applications and reviewers. But the reality is that correlations, you can have two data sets that both have 95 samples, but they have very different sampling strategies and therefore very different correlation structures, right? So I don't use it as a hard and fast rule, basically. There's many different kinds of sampling strategies, and I think this is an area where there's, I think there's exciting things happening. So here, I'm a neuroscientist, so I illustrate with brains. This is one type of sampling strategy where you're analyzing one particular region from many different individuals, right? So now you have all these sources of variation that correspond to, the reality of dealing with samples from different individuals, right? All the things that I mentioned earlier. Here's another kind of sampling strategy where you're looking at multiple regions from one individual, right? Now you're controlling for, differences in age, gender, cause of death, ethnicity, whatever, and regional variation is your major biological source of variation. Here's another strategy. This is from the Allen Institute. In fact, you might recognize this. I think this is from the macaque data set, but it's basically a laser microdissection of one slice of macaque cortex where they're using a laser to cut out different pieces spanning the cortical wall, right, from the bottom to the top of the cortical wall. And then more recently, this is single cell analysis, right? It's so hot right now. So that's the latest source of, or the latest kind of sampling strategy. In my lab, we've developed an alternative sampling strategy that we like a lot, which is illustrated here, where we take one tissue specimen and we cut serial sections from one end to the other. And we extract RNA from every section, and then we do a co-expression analysis. And that has a lot of advantages in that it controls for many sources of biological variation. But compared to single cell analysis, it has, I think, some attractive attributes. It's stereotyped, and you don't have to beat up the cells, and you preserve spatial information, cellular stoichiometry, et cetera. I'll talk more about that in the last talk. So Steve mentioned this a little bit earlier. This other kind of big question is, are you analyzing one biological cohort or more than one? Are you trying to do a differential network analysis or just a straight up co-expression analysis? Differential network analysis, I think, is really cool, and you can learn a lot of interesting things doing it, but it can also be a little bit tricky. And if you're designing an experiment from scratch, there's different ways you can consider both structuring the data generation and the data analysis, and there's not necessarily one right path, right? This module preservation function that Steve and Peter have created is one very helpful tool for looking at differential co-expression relationships. You have to be careful when you're doing that the data sets you're comparing have comparable numbers of samples, ideally, right? Otherwise, it's something you have to control for explicitly. So, Peter mentioned this earlier as well. There's a whole cottage industry devoted to, identifying clusters or modules or patterns in data, and there is nobody can claim optimality, right? But I think we're all here because we've heard of WGCNA, and with good reason. I think it's become kind of an industry standard, and in large part thanks to Steve and Peter's, devotion to maintaining, tutorials and documentation in the RPAC. It is to me the most obvious place to start. And also, you guys are probably aware, but if you just Google gene co-expression, I'm pretty sure that Steve's website is still the #1 hit. And on the website, there's all of these tutorials for previously published studies, and you can literally download the exact same data that were used by the authors, download the tutorial, copy and paste the code into an R session. Even if you don't know how to program in R, you can still see just by trial and error, cause and effect, what the code is doing. So it's really a great resource. This is another topic that has been already touched upon a little bit, but the reality is that despite our best efforts, it's there's a bit of a song and dance that happens, I think, in a lot of publications, and that it's palatable for the reader to present one co-expression network from a given data set. But the reality is that you can build many co-expression networks from the same data set. The core signatures will recur over and over and over again, right? But at the same time, it can be difficult to decide how many modules is enough, right? The parameters in WGCNA that have the biggest effect on that are the deep split, but also the minimum cluster size, right? If you try building a network with only, 3 genes in a module, you're going to get a lot of modules, right? This is an example of 3 different networks built from the same data set. So here again, I'm clustering the eigengenes, right, based upon their similarity to each other. And you can see here the settings were such that we identified a pretty manageable number of modules, right? Now, here we're fragmenting the tree, right? Same data set, but we're finding a lot more modules. And now here's a third view where we find even more, right? And you can kind of see, just from looking at the eigengenes below, for example, like this one right here, now it's over here, and now it's over here. So it's like things are fragmenting, right? And you don't know at what point you've sort of left biology behind and you're just splitting hairs, right? And you might think, well, this is too many modules. But then you can see something like this, right? This one module in here that I've tried to highlight. And when you look at the genes in this module, you can see all these mast cell genes, right? So that's it's seldom this obvious, right? But once in a while you find a module like this. So the top 15 genes, almost all of them are related to mast cells, right? So this is a module that wasn't present in the first, 2 levels of the network that I showed you earlier. So in some ways, what I'm saying is that you need to have biology be your guide, right? If you're interested in mast cells and you're looking for a signature of mast cells and they're not a well-represented cell type in whatever system you're studying, but that's the system you need to study, you might need to build a network that has a ton of modules in order to find it, right? So that's just something to think about. There are statistics that you can use to sort of help guide this process, and I know Steve has published on this, things like the silhouette width and modularity. They look at the density of modules, the separability of modules, the stability of modules. I've tried to work with those to some extent, and I've often found them to be good ideas that are of limited utility in practice. That's just been my own personal experience. Again, I think that biology should really be your guide. And when you're looking at, for example, a new biological system or looking at it for the first time through the lens of co-expression, you're never going to be able to explain every single module that you find or even half of the modules that you find. Most of them you're not going to be able to explain, at least right away. So Steve's advice, to focus on the low-hanging fruit is, I think, very good. You want to be able to explain the largest, most robust patterns in your data. And if you can't do that, it's going to be a lot harder to explain things that are smaller and harder to find, in general. So in my lab, when we try and characterize modules, it's really a two-step approach, and one step it's disproportionate in that the first step is really where you put most of your energy, and that's doing enrichment analyses, right? So there's huge compendia of biologically informative gene lists that are out there now. Some of these you can access through the WGCNA package, so like these brain lists, blood lists, immune pathway lists, et cetera, right? These are lists of genes that all have relevance to those particular biological systems. There's a gazillion databases out there, right? So depending upon which area of biology you're working in, you should know probably where you can find the most informative gene sets. This one, Molecular Signatures Database, is kind of nice because it's sort of a repository for many different sources. I think they're up to over 10,000 gene sets now. That it encompasses gene ontology. It encompasses a lot of the pathway databases, chromosomal cytobands. It also has curated gene sets from published studies, and you can download this whole database in an XML file, and you can read it into R, and you can parse it. It's a nice thing to work with. We use the Molecular Signatures Database and our own collection of gene sets from a number of different sources, and when we build a co-expression network, we can, in an automated fashion, do these enrichment analyses for a large number of gene sets, and I'll just show you how that can give you sort of a sense of the structure of a network. This is a co-expression network that was generated from a particular part of the human brain called the superior frontal gyrus, and which is highlighted in red, kind of cut off at the top. And this is from about 40 different individuals. And again, we're clustering the module eigengenes. And when we look by enrichment analyses, this is a set of genes that have been experimentally validated by the Allen Brain Institute on the basis of in situ hybridizations in the mouse brain to be expressed specifically by oligodendrocytes, right? And on the y-axis, that's minus log p-value. So it's basically reporting the enrichment results using a Fisher's exact test for this gene set in all of the co-expression modules in this network, right? So you can see there's this blue, light green, and purple that really have strong enrichment. Here's a different set of a different gene set, but it's also an oligodendrocyte gene set, right? So this is from Ben Barris's lab at Stanford, and in this case, he's purified homogeneous populations of cells from the adult mouse brain, done a differential expression analysis, said here is my best set of oligodendrocyte genes, and it shows the same pattern. Here is another oligodendrocyte gene set from Nat Heinz's lab. This was produced using this TRAP method, this translating ribosome affinity purification method. But you can see it hits the same modules. And then this is one that I produced from the Nature Neuroscience paper that Steve mentioned in 2008, where I found a co-expression module corresponding to oligodendrocytes in human brain, right? So it's very consistent, which is nice. Now you can see that on the left part of the tree, those modules are enriched with markers of neurons, right? From the Allen Brain Atlas, from a single cell study, from work that I did, and from the Barris lab, right? So you can see this whole bifurcation in the tree, the whole cluster at left is really modules consisting of genes that are primarily expressed in neurons, right? So you can start to get a sense of the structure of the overall network. Over here, you can see there's a green module that's enriched with markers of astrocytes, which is another major cell type in the human brain. We can see that with multiple independent data sets. And then over here, I only have one gene set for this module, but that's basically a microglial module, right? So again, if you have a collection of informative gene sets, particularly with respect to your biology, it's a great way just to get a lay of the land, right? Okay. So the second way that we approach module characterization is the low throughput way, which is histology, right? The question I always ask myself when I am looking at a biologically interesting co-expression module is, which cell type or cell types are likely to be expressing these genes? That's kind of the point of entry for me to try and understand any module. And when you're dealing with postmortem human brain, histology is even more low throughput than it is with model organisms. But we benefit from these giant resources that have been developed over the past few years, like the Mouse Brain Atlas from the Allen Institute. If you study mice, there are other databases like URExpress and GenePaint that have, basically whole body in situ hybridizations for most genes in the genome. So those are great places to start. Here's an example. This is a module from a collection of samples from actually infant human brains from a variety of regions. And you can see that it's just, this module consists of genes that are spiking in just a handful of samples. If you look at the top 3 genes ranked by KME, and you go to the Mouse Brain Atlas produced by the Allen Institute, which has in situ hybridizations for every gene in the mouse genome done in serial sections through the adult mouse brain, you can see that this first gene, F5, is blazing in this particular cell type called the choroid plexus, right? This is the cell type that produces CSF. The number 2 gene, CLIC6, also expressed in choroid plexus. Number 3 gene, this is a orthogonal view of the brain, but you can see that's the choroid plexus right there, right? So that's helpful if you have access to a resource like that for your biological system. Sometimes you find modules that look interesting but don't show any really hit you between the eyes, enrichment p-values, or cell type associations. And I would say that here, what you want to have is independent validation. I mean, ideally you want that for all of your modules, right? But if you see the same module or very, very similar modules 2 or more times in independent data sets, the odds of that happening by chance are vanishingly small, right? So that's real biology, and if it's unannotated, right, it's potentially very novel and interesting biology. Yes?
Are you going to go a little bit more into cell type specificity for this? I tried to use comparison data set before to tease out neuron versus actual site evidence. What is the name of that?
Yeah. I will talk about that more in the next part of my second talk, I guess. With Ben's data, you might try thresholding it at a higher fold change because they have these supplementary tables, and I think they have a pretty, the tables include genes that have like a 2x fold change, whereas we truncate the definition of the gene set at 10x fold change, which is just a more conservative way to do it. Yeah. But I'll talk more about that in the next talk. Other questions? Okay. Oh, well, that was the last slide. Yeah, so this is a book chapter that I wrote in a collection of book chapters arranged by Giovanni Coppola, who's going to talk, I think, this week, and touches upon many of the same points that I made today. And I think I included it on the website. Hopefully you can download it. So, yeah.